Okay, in chapter 7, we're going to continue talking about population genetics. And if you remember in chapter 6, we sort of laid the foundation for population genetics or evolutionary genetics, looking at how evolution can change those populations. And we specifically looked at what happens to the Hardy-Weinberg equilibrium or the Hardy-Weinberg model when we introduce mutation and different kinds of selection. For chapter 7, we're going to continue doing that same sort of thing, but now we're going to look at what happens um, when you have migration, what happens when you have genetic drift, and that's typically something that is most pronounced in smaller populations, and then also what happens when you have non-random mating. It's always nice when we're sort of talking about these theoretical things or mathematical things like we saw a bunch of last chapter in chapter 6 to sort of couch them or look at them in real life situations. And so and here what I want to do is I want to take a natural population and then sort of introduce you to this natural population and sort of an event or events that have happened in these natural populations, and then we'll jump back into looking at sort of like the theoretical stuff and see if in fact what we are predicting from the theoretical side can help us to understand what's going on in the natural population, and more importantly to help us recover sometimes natural populations. And a great example of this is the greater prairie chicken. Here this is a picture of a male greater prairie chicken. Look, they're other names for them. They're a type of grouse. So some of you, we have grouse here in Utah. Some of you, can see the similarities between this. This is a separate species. Historically, these were very successful species. There were hundreds and thousands of them across the Midwest, specifically in Illinois, and that's the what we're going to look at sort of in more detail. If you were to go back 200 years ago, the entire state was covered with them. One of the more impressive things about these chickens is their mating rituals. The picture before was that of a male performing a mating ritual to going through the male sexual display. Some people have called these prairie chickens 2-pound chickens with 10 pounds of display. They do pretty elaborate sort of dance when males gather and sort of compete with each other to show the females just which males were sort of the sexiest. And here's a link on YouTube to a video that kind of shows you that their mating ritual and their sexual displays. It's pretty impressive, and that's if you had heard of the greater prairie chicken before, probably you had seen it a documentary or something that shows this display. This is a table that shows prairie chicken numbers throughout sort of a timeline in Illinois. As you can see, in 1933, estimated about 25,000 of them, and we've had biologists going out and making estimates of population sizes here in the U.S. for a long time. And so biologists back in the 1930s, this is probably a typical number of prairie chickens, 25,000. So a pretty good chunk of population, pretty good population. But what we see in the 1960s, 1970s, 1990s, and then in the mid 1990s, we see a dramatic decline in the population numbers of the greater prairie chicken. So much so that they were on the verge of extinction in the mid 90s in Illinois. And so we can look at that, we can look at those data, and then we can say, okay, what's going on here? Something had to have occurred, and not surprisingly, in the last 100 years or so within Illinois that has dramatically decreased the prairie chicken population. I'm sure many of you have been out to the Midwest or driven through the Midwest, or maybe even grew up or have lived in the Midwest. And usually, when you ask people who've gone out to Iowa or one of those other Midwestern states, what their impression of, they usually say, oh, lots of farmland, miles and miles of really flat and lots of farmland. But obviously, this wasn't always the case. It was the case that for a long time before white colonists came in and started to farm the land, and even after they first began to come in, the farming didn't really kick in until John Deere invented the first commercial plow in the 1830s. And the reason is that the prairie, the native prairie out there, formed a very, very thick and hard sod. So sod, we think of sod, if you've done any landscaping, is just basically rolls of grass, right? But the sod in the prairie, it's made up of the same sort of thing. If you look at sod in landscaping, you see the grass, and then you see a bunch of dirt and a whole sort of a thick mat of roots to the grass underneath the grass blades. And then the prairie sod is similar, but on a much larger scale and a much harder scale. And so for a long, long time, it was not opened up to large, intensive farming because we just didn't have, with a wood plow, you wouldn't really be able to get in there and chop up and dig away the hard compacted sod. And even with some of the other plows made of metal, it wasn't really until we developed a still, which is has a very hard type of metal alloy, that we're able to actually go in there and start breaking up the sod and actually convert this very, very, what ended up being a very, very fertile soil or fertile place with lots of fertile soil into an area that now is sort of the breadbasket for the world and has large scale farming that feeds lots and lots of people. But so that was from sort of human population development perspective. That's good, but for the native species that had been there for thousands of years, right, living on the prairie and evolved and adapted to the prairie landscape, coming in there and all of a sudden converting large stretches of land, what was originally prairie, into farmland, had very negative effects on some of the native species. And one of the native species that was really impacted by it, obviously, was the greater prairie chicken. Here you see the state of Illinois through time, and the greater prairie chicken population is there in blue dots superimposed on an outline of the state. If you look back at 1810, 1820, you can see, just like we said, that the state was pretty much covered with greater prairie chickens. But what happened after 1820, 1830s, the still plow, you open up a lot of the land in Illinois to farming, and within a little more than 100 years, you can see a massive reduction in the habitat of the greater prairie chicken, as sort of represented by the populations that were still around. And to be fair, this, even back in the 1930s, people were like, oh, this is they noticed this, and it concerned some people, and in fact, they passed a hunting ban on the greater prairie chicken because you were destroying the habitat through farming, but then also people, like grouse and pheasants today, people like to hunt them and eat them, and so they decided to ban hunting in the 1933s. Fast forward in 1933, fast forward a few decades, in 1962, you can see that it was still kind of, still the prairie chicken populations were going down. 1994, you only have now 2 prairie chicken populations in the entire state of Illinois. And those 2 locations you see there in the map of 1994 were set up as sanctuaries in one particular county, Jasper County, and that was the only place left in the whole state where you could actually find greater prairie chickens. So you can see this is a dramatic decline in prairie chicken population size, populations in Illinois throughout 100, almost a 200-year time span. But we can also look at it and say, okay, so in 1933 we banned hunting. Obviously, there was more going on in the population than just to the population decline than just pressure from human hunting, because that did not seem to to cure the rapid decline of the prairie chickens. This graph shows the chicken populations in the sanctuaries after during the 1960s, and what we found is that in fact when we actually protected the chickens, set aside habitat for them, they initially responded fairly well to it. In 1970, you had the population grow to 200 prairie chicken males, prairie chicken cocks, right? But even though then you see a dramatic decrease from 1970 to 1980, continuing through the 90s. And so you have to say, okay, what's going on? Did something happen? Externally, no, nothing really happened. We decided, okay, we're going to preserve this habitat for them. We're not going to hunt them. Why didn't the prairie chicken populations respond, right? Why didn't they go to 200 and either just continue at 200, right? Or, if that was sort of the carrying capacity of the sanctuaries, or why didn't they increase beyond 200? But instead, what we see is they actually decrease from 200. And then in the mid 1990s, you see a bit of a rebound to where, from where they were pretty much almost going extinct in Illinois, right? And so what had happened? How did we change the course back in the 1990s? And so that's what we're going to do. What happened in the 1990s is we began to approach the conservation of this species from a population evolutionary genetics perspective, and from what we learned theoretically from what we're talking about, right? From the Hardy-Weinberg equilibrium, Hardy-Weinberg models, and the assumptions of the models and the patterns of what happens to populations when those assumptions are violated. And so this shows you that we're not just going through this theoretical exercise just to go through a theoretical exercise, right? Just because we want to, but actually there are real world implications of the stuff that we learn from this, and especially when we start talking about endangered populations and conservation. These have real world impacts. Okay, so we're going to leave the prairie chicken story there, come back to it after we jump in to looking at migration, genetic drift, and non-random mating. And hopefully this will help us to understand why, in fact, these populations did decline. Okay, so the first thing that we have to do in the discussion of migration is sort of define it in evolutionary terms. Usually when you think of migration, people think of the seasonal movement of individuals. For instance, birds flying south for the winter. From an evolutionary biology perspective, that's not what migration is. Migration is the movement of alleles between populations. So it's genes moving from one population to another population. We call this gene flow, and it can take the form of individuals moving from one island to the next and then mating with individuals on the new island, or it can take the form of pollen being blown in the air, pretty great distances. It can also be, for instance, sea urchins broadcasting their gametes into the water column and then traveling, again, pretty far distances on the ocean currents. Here is a diagram of, usually when we start talking about immigration, we try to set up a system that we can kind of account for things sort of a theoretical framework, and a typical theoretical framework in the discussion of migration is island to continental migration. And here we see a continental population, big, right? An island population, not so big, small. And the arrows indicate the influence of the gene flow between the two locations. And so the big arrow is indicating that the continent can have a big influence on the island population because it represents a large portion of gene flow, right? Because the island's small, whereas the island isn't going to have much influence on the continental population in an evolutionary sense because it will probably generate only a few migrants onto the continental population. The continental population is large. So let's look at this from the Hardy-Weinberg framework. Start up in the sort of upper left-hand corner. You have an island population. On that island population, not the island population, but we have in the island and the continental population, we have two different alleles at a particular locus or gene. We have an A1 allele and we have an A2 allele. We start with the island population at fixation, meaning the entire island population has an A1 allele, so all of the individuals on the island are homozygous for the A1 allele. All right, they mix, they randomly mate. When they randomly mate, since all of the alleles are A1 alleles, even with them randomly mating, we only have one particular potential genotype, and that is A1A1, and this is in Hardy-Weinberg equilibrium, right? Okay, so they, if those eggs get fertilized, they produce zygotes. The zygotes grow up, form 800, I guess in this case, mice, right? Then we have the migration event. The migration event is a group of 200 mice from the continent coming into the population, coming into the island population. I guess the mouse and island isn't that great of an example, but let's just go with it. So you have 200 individuals, and those mice have A2. They are homozygous for the A2 allele. And so now you have 200 individuals, right, on the population that are A2, 100 individuals that are, 800 individuals that are A1. Your new allele frequency now is 0.8 A1 and 0.2 A2. So pretty straightforward, right? Big migration is going to change your island population, and that is going to take you away from the Hardy-Weinberg equilibrium. So migration can affect the Hardy-Weinberg equilibrium and take you out of the Hardy-Weinberg equilibrium. So if we set up the mathematical treatment of migration in the Hardy-Weinberg model, and you can read about that and look at it in a little bit more detail in box 7 in the book, we would generate an equation such as this one. P sub I prime, right, equals 1 minus m times P sub I plus m times P sub c. And so let's define some of those variables. So P sub I is a frequency of the particular allele on the island. So let's just say take A sub 1, A1 like we were looking at before. So what's the frequency of A1 on the island? That would be P sub I. P sub I prime is the next generation frequency of that same allele on the island. So, okay, how much, what is there, what in that one generation, what has happened to that frequency? P sub C, as opposed to P sub I, the C is continent, I is island, P sub C is the frequency of the island on the mainland. So all those are pretty straightforward definitions. And then here's the important one, M, which is the fraction of the island migration that came, the fraction of the island population that came from the mainland. So that's basically an immigration rate, right? And so if you have a high immigration rate, then you're going to have okay, so let's just look at the equation. P sub I prime, so what's going to happen to that? That's going to go up, right? If you have a large immigration rate, M, right, because that's going to be influenced by the allele frequency in the continental population, right? 1 minus m means that that's going to diminish the input of the native island population on the frequency of the next generation. Okay, so the important thing is, let's look at how, if we can model how they change from one generation to the next, or the delta P, right? What is it? And then more importantly, say, okay, when ultimately, because we want to make predictions in evolutionary biology, ultimately, when are they going to reach an equilibrium, right? What's going to happen? Where is that equilibrium going to be reached? And how long is it going to take for it to reach it? Let's make these evolutionary predictions. And so what we find is that when we set delta p sub I equal to 0, so p sub I, so you can then you can go through the rearrangement, the algebraic rearrangement if you want, but we get to the equation m times pc minus p sub 1 equals 0. That's the equilibrium point. And I don't want you to worry too much about the math, but just look at that that reduction in the equation and see what it tells us. So basically, this is telling us that the frequency on the island will remain the same, will remain constant when m is equal to 0, right? When there's no migration. So m equals to 0 times pc minus p sub 1 is obviously going to be 0. Or within the parentheses, p sub c is equal to p sub I when the island and the continental populations have the same frequencies. So this is important because that sort of tells us what immigration does. Immigration makes populations, when there's more immigration, it makes populations. Well, so immigration is going to continue, if immigration continues, it's going to continue to shift or cause the populations to evolve until that frequency is reached. Okay, once that frequency is reached, then you're not going to have changes in allele frequencies each generation. And so what migration or gene flow tends to do is tends to make populations become more similar. And when there's a lot of migration between populations become more similar quicker. When there's little migration between populations, it takes them a lot longer to do it. And so that's sort of the overall take-home message of that. So we'll leave sort of the theoretical stuff. Let's now kind of start talking about some real-life examples. Okay, we've seen what happens when you have two populations and there's constant migration between the populations, or I guess more specifically, we'll look at what happens when you have a continental population and an island population, and there is gene flow or migration to the island from the continent. Eventually, all other things being the same, the island population should show the same frequency as the continental population. But here we want to look at, obviously, real life examples can be a little bit more complicated. For example, what happens when the different populations are under different selective pressure? King and Lawson did a study on the migration of water snakes in Lake Erie to islands out in Lake Erie. And they wanted to look at this, to look at how migration was affecting the composition of the populations, and then also how selection, or is selection, would act differently in it, and therefore produce different populations with different frequencies of a particular characteristic. The characteristic they were looking at were the banding patterns in these water snakes, Nerodia. Here you see a picture of the three different morphs. You have a sort of a strongly banded morph or form of the snake, you have an unbanded, and then you have an intermediate banded snake. These snakes here are hanging out on some limestone rocks, and so if you just sort of by looking at it, you might think, okay, predators searching for these snakes on the limestone rocks would probably see the banded snake here first before the unbanded. The unbanded is probably more cryptic in this sort of limestone environment. So how does this play out on the islands in Lake Erie? So here you have a map of Lake Erie. You see to the north is Ontario, Canada. To the south is Ohio. Um, they went and sampled and looked at snakes on the islands and on the mainland, both the mainland in Ontario and down here in Ohio. They have a peninsular mainland population. And what they found is that in the mainland populations, the snakes were almost exclusively banded. They were almost all of them were banded. When they looked at the island, they saw that the different islands, they saw that most of the snakes were unbanded. There were still banded snakes out there, but most of them were unbanded. And what they noticed was that on the islands, the islands were made of these limestone rocks. When they would bask on the limestone rocks, especially the young snakes, the banded uh, snakes were much easier, like we said before, were much easier to spot, and the predators they assumed would therefore be much more likely to capture those young snakes. That wasn't true on the mainland. The mainland had a different sort of environment where the banding likely helped in the snakes in being more cryptic. So here we have this classic sort of continental populations, island populations. Looking at the different island populations, you see that you have a constant migration of these water snakes out to the islands, and so you have to say, okay, if the big populations on the mainland are all banded and there's this constant migration out to the islands, how come all of the populations out on the islands are not also banded, because you have these big continental populations. And so the apparent answer is that, okay, well, it's a little bit different environment, and so they're going to be under different selective pressures. And so the selective pressure out on the islands, and so this is all sort of sounds good, right? This is all good in theory, but what you want to do is you want to then go out and look at it and see if in fact you can make sense of it from sort of real life data. So these are the data that King and Lawson collected. As you can see there on the left, on the peninsular mainland in Ohio, and then in Ontario, they're almost exclusively banded. And then the three different sort of complexes of islands: Kelly's Island, Bass Complex of Islands, and then the Middle and Pelee Islands, I guess is the way that you say that. You see that you don't get that same distribution. Yes, you do get banded and sort of intermediate ones. But if you look at the A category, which is not banded, completely not banded, you see that especially in the Middle and Pelee Islands, you get a higher. That is the most frequent. That is the most frequent type of snake you find out on those islands, and so we can go back and look at the previous slide, and you can see that the Middle and Pelee Islands are actually kind of out there in the middle of the lake, and therefore would have less likely have less migration out there, with probably with some strong selection for the unbanded snakes, whereas the other islands are probably getting more migration. And with that larger amount of migration, you have a higher amount of the genes that code for those banding patterns, which therefore influence the intermediate banding patterns, and so you don't see quite as many unbanded snakes there. But again, here we now see there's this balance sort of between selection and migration. But we should ask, okay, what would happen then if selection stopped? So all of a sudden, or not, yeah, all of a sudden, if selection stopped, say the island composition became different, you would then expect what to happen? Hopefully you've thought about it, and you would answer that, okay, that you would expect the amount of banded snakes on the islands to then approach the frequencies that you see on the mainland. So I'd like to point out that as we've looked at the Hardy-Weinberg model and its assumptions, as we've been doing this, we are also kind of just looking at how different things can cause populations to evolve. So we're looking at the Hardy-Weinberg model, its assumptions, but we're also looking at evolution, how different things can cause populations to evolve and what kind of patterns are generated in that kind of evolution. So up until now, we've looked at mutation, how mutation contributes, how different kinds of selection contribute, and how migration all affect evolution. Now we're going to look at another phenomenon called genetic drift. And genetic drift deals with the part of the Hardy-Weinberg model that says populations have to be large or infinite. When populations aren't infinite or very large, and as they become smaller and smaller, we see the effects of what we call genetic drift. We've also up until now been really good at sort of explicitly underlining the fact that natural selection is a non-random process, right? And so there's explicitly non-random processes involved in evolution by natural selection, but there are also some random processes. One of the random processes was something we've already talked about, mutation. Mutation is what nucleotides get changed and what amino acids then result from that change is a random or mostly random process, but which mutations are then seen as favorable by natural selection or unfavorable by natural selection is a non-random selection of that, right? And so here we're coming back and looking at some random stuff. So if you have a population, let's just take a diploid organism, and they can even be randomly mating, and they're going to be producing gametes that are then going to fuse and produce your zygotes, and then your zygotes are going to become your individuals of the population. We can describe all of the potential gametes generated by a population. When I do this, I think I mentioned this before, I like to think of sea urchins. Sea urchins sort of broadcasting their gametes into the water column. And all the sort of the sum total of the potential gametes that are going to contribute to the formation of the next generation is what we call the gene pool. And so here's a gene pool. In a gene pool, we can describe a gene pool by the allele frequencies. And so again, we're going to stick with the alleles A1 and A2. And in this particular gene pool, if we went through and we counted up all the A1s, we'd see that 60% of them, and I don't know if that's actually true in this diagram, but let's just say it is, 60% of them are A1 alleles and 40% of them are A2 alleles. Okay. Then here's our gene pool. In the next generation, we are going to select out, we're going to sort of reach our hand into the gene pool, pull out a gamete, then pull out another gamete, form a zygote, and that's going to be our individual. And so here with our allele frequencies, and we've done this before in chapter 6, we could go through and we could form our genotypes of the next generation. If we select a large enough population, we're pretty much, for the next generation, we're pretty much guaranteed that we would create a population that is in Hardy-Weinberg equilibrium. Okay, but what happens if we don't? What happens if we select a small population? What happens if we only, instead of pulling out gametes and forming 1,000 individuals in the next population, what happens when if we were to only select 10 individuals for that next generation? That is where genetic drift comes into play. Okay, so let's do this with a theoretical population of mice. We're going to start up here on the top left corner. Again, the allele frequency is 60% A1, 40% A2. In our gene pool, we're going to reach our hand in, grab one gamete out, and let's say it's an A1, and then the second gamete is an A2. We therefore have made an A1, A2 individual. Let's say we do it again, pull out A2, A2. Now have one A2 homozygous individual. Let's do it again and pull out an A1 and an A1. We now have a homozygous A1 individual. And we go through and we do that for 10 new individuals. And there on the right, you see that we end up pulling out and forming 6 individuals that were A1, 2 individuals that were A1, A2, and then 2 individuals that were A2, A2. Those individuals grow up to be juveniles. They then grow up to be adults. And if we then look at that population and we calculate an allele frequency, what we'll find is that the new allele frequency in this next generation is 70% A1, a 70% frequency for the A1 allele and a 30% frequency for the A2 allele. So the A1 has gone up and the A2 has gone down. This, again, a change in allele frequency is evolution. But how did this happen? It was just random sampling error. It's an easy way to sort of think of genetic drift. Genetic drift is just random sampling error in the formation of gametes from one generation to the next. Could we do it again? What would happen if we were to do it again? We could go through, we could pick them out, we could pick out the gametes again, and it's very likely that we're not going to end up with a population made up of 6 A1 homozygous individuals, 2 heterozygous individuals, and 2 A2 homozygous individuals, but something different. And if we could do that a whole bunch of times, and the next slide is the result of doing that a whole bunch of times, a distribution. Those of you who've had stats will notice this is a pretty good approximation of a normal distribution. And so there in the middle is the where we see the new frequency of the A1 allele at 0.6, right? So where it doesn't change, and we can actually take the area of that particular sort of tan bar, put it over the total area of the distribution, and that gives us that 18%. All right, so there's an 18% probability that the population in the next generation stays the same. There's also a probability that the allele frequency of the A will go up, or the allele frequency will go down. You can go all of the way to, and there is, could you potentially, in the formation of those 10 individuals, for the 20 gametes that you are going to use to form the 10 individuals in that next generation, draw all A2 alleles? Sure, there's a probability of that. But it's a small probability. Is there a probability that it's all A1 individuals? Sure. It's that also is a small probability. So what you can see is genetic drift, and that's why you get the drift part of it, is it's sort of random because it could go up or it could go down or it could stay the same. And in one generation is most likely that the allele frequency isn't really going to change that much. But if we look at what happens over multiple generations, that's where that drift can actually make some significant evolutionary, or can produce significant evolutionary outcomes. So there's a mathematical description of the extent of the deviation or the sampling error, and this equation, you don't have to know this equation, but I want you to be able to kind of look at it and say, okay, this is why large populations with large population sizes we get small deviations or small sampling error, and small population sizes we get with large we get small, and with small population sizes we get large deviations. So the equation is sigma equals the square root of p times q over n. So looking at this equation, we see that two things affect the shape of that deviation, the extent of that deviation, or if we were to go through and do a complete distribution, these are the things that would affect the shape of that distribution curve. One is population size. So when we look at the equation, we see that n is on the bottom, and so as n gets larger, right, that means that sigma is going to get smaller. As n gets smaller, that means that sigma is then going to get larger. Also, the values of P and Q, these are the allele frequencies. And just in general, and that's what this little box shows, just in this little box with the multiplication in it, shows that as the allele frequencies are more similar with each other, for instance at 50% and 50%, you get larger deviations than when they are more dissimilar. So they're at the bottom, 0.9 times 0.11. Those are the two things that actually affect it. But that is just, that's the deviation, right? That's the deviation. And that explains why you can also use this equation to think of a coin flip. A coin flip, you have a 50% probability of getting a heads and a 50% probability of getting a tails. That's that top one. 0.5 times 0.5 equals 0.25. If you were to flip the coin, and you could plug this in and do the math. If you were to flip the coin and flip the coin 3 times, your end value would be 3, right? And so you would have 0.25 divided by 3, take the square root of that. That would be a fairly large deviation, or much, much larger, and that's why you could flip it 3 times and you get heads, and nobody's going to say, oh, what's wrong with this coin? This must be a trick coin, right? Because you expect a large deviation with that small sample size. But if you were to flip that coin 200 times and you got all heads, right? N then becomes 200, right? And so you would expect small deviations. With those small deviations, you've actually had a large deviation, right? You're now towards the end of that distribution, and you would say, okay, something's wrong with this coin. And so that's just sort of intuitively we understand, or we can connect this population size effect on the extent of deviations. This is just a mathematical, numerical way of doing it. The next slide, I'll show you a graphical way to interpret it. This just shows you graphically what I just explained. So on the top, you have your large population, right? Sigma there is that deviation you see. In a large population, you get a small deviation. In a smaller population, you get a larger deviation and a larger sort of spread to your distribution. Okay, so let's take another theoretical example. Here we have a population. We're going to use big A and little a as the alleles. We have a population of 100. With the allele frequencies equal, we have equal number of big A homozygous individuals, 25, little a homozygous individuals, 25, and heterozygous individuals, we have 50. But each of these individuals is going to so then each of those individuals is going to produce lots and lots of different gametes. Let's just pretend we have that gene pool, and now we're going to form another population in the next generation with 100 individuals. Okay, so the probability that we reach our hand in and draw out a little a, and then one little a, and then another little a, if it's 50-50, the probability of pulling out a little a is 1/2, right? The probability to reach in and draw a second A is also 1/2. The probability of doing both of those events, one event and the other event, in statistics, whenever we hear and, we multiply, is 1/4. And so we could actually calculate the probability of if in that next generation we were to draw out all little a gametes, or the next generation of 100 individuals would result in all homozygous A individuals. And we can, down there at the bottom, we see that probability would be 1/4 to the 100th power. And so there's very, very, very little chance. It's not impossible, but there's a very, very little chance that you could do that. Okay, so that's just showing you how you could do that mathematically. So let's look at not such an extreme case. Let's just look at calculate what the deviation would be for a large. We'll just take sort of just randomly take a large population and a small population, and look at where it's likely for that, where our allele frequencies are likely going to venture in that next generation, comparing a large population with a small population. So here are two theoretical populations. Our large population, we're going to say has 5,000 individuals, and our small population, we're gonna do sort of an extreme example, and let's just say that next generation only has 2 individuals. We'll take that equation that I introduced a couple of slides ago. Again, this frequency is the frequency of both alleles, P and Q, or A1, A2, or big A, little a, are both 0.5. We plug that into the deviation, the standard deviation equation, sigma equals the square root of 0.5 times 0.5 divided by 2 times 5,000, because there's 5,000 individuals. That means there's 10,000 gametes, right? And we get a deviation of 0.005. And so what that's telling us, that it's most likely you're going to see a deviation in the next generation, but if we go up 0.005 or down 0.005, right, that means that we are going to we're most likely going to be somewhere between 0.495 and 0.505. So it's not going to move all that much in that large population. It's likely not going to move all that much. In the small population, we do the same thing in the equation. We times it out. We end up getting our standard deviation, sigma, equal to 0.25. That means that it is likely that it's fairly likely that it is going to deviate quite significantly from that 0.5. So let's just say we'll take the larger one. It goes to, let's say in that small population, it goes to 0.75, right? We'll take that one, and then we'll look at what a couple of generations of doing the same thing, what's likely to happen to that allele. So now we're looking at producing the third generation. So the second generation now has 2 individuals, right? They're still, producing a whole bunch of gametes. But now the allele frequencies are different. Now P is equal to 0.75, or we'll say A1 is equal to 0.75, and A2 is equal to 0.25. We plug that again into the equation, and we now have a standard deviation of 0.22. So we can add or subtract that from 0.75, and that tells us that we're likely to go back down in that next generation towards 0.5, or we could go up, and in fact it wouldn't be that out of the ordinary that we would get all the way up to 0.97. Another if that happened again, or even there is even a chance here, and not that small of a chance, that you could have the A1 allele going to fixation in a matter of 2 or 3 generations with that small sample size. Fixation is when we say fixation, we're talking about an allele going to a frequency of 100% in a population, or the opposite allele going to a frequency of 0%. It becomes fixed. That gene becomes fixed for that version of that gene, that allele, at 100%, or it gets weeded out of the population. This slide shows you a whole bunch of simulations for populations of different sizes, starting out at the having an allele frequency of 0.5. Up there on top, you have a population size of 4 compared to on B 40 compared to C at 400, and each of those is separate sort of simulation runs. And so what you see, and this is just sort of graphically showing you what we were just talking about. With a population of 100, you don't get that, or the 400 there on the bottom, you don't get a lot of deviation from one generation to the next. It's kind of slightly up, slightly down, slightly up. And after 100 generations, you still haven't gone to fixation. You have some populations that have A1 frequency of the A1 allele up around 80%, and other populations down there around 25%, but you still have both alleles present. In the population where the up top in A, where your population size is 4, you've gone to fixation for all of your simulations by generation 20. And then the B, where population size is 40, you have some populations that have gone to fixation and some that haven't. The other column, I don't want you to worry too much about that, but another effect of genetic drift is a reduction in the frequency of heterozygous individuals. And so you see here in the first simulation, after 20 generations, you have, the vast majority of your populations going to fixation. When you go to fixation, right, you no longer have heterozygous individuals. And so that just shows you that sort of that thing. So we can view genetic drift as sort of this constant march towards homozygosity that's more pronounced in smaller populations than large populations. A way to remember it, or an analogy for this, is to think about your population out there on the end of a pier, right? And there's a bar out there at the end of the pier where sailors go and drink at night, and they need to walk back to the shore down the pier. And so you can think about the population size is directly correlated to the amount of alcohol the sailor drinks. If the sailor drinks a lot, it's sort of inverse. But if the sailor drinks a lot of alcohol, that represents a small population. So the sailor is going to leave the bar and be taking dramatic steps to the left or to the right or to the left a couple of times in a row, and it's a high probability that the sailor will fall off of the pier before he makes it to the shore if he's drank a lot, or if you have a small population. A large population would be a sailor that drinks, soda or maybe not very much alcohol. Small deviations for each step, and each step is a generation. So he leaves the bar, each step is a generation. With large population size, that means there's very little was drank, and so there's a very small probability that the sailor's going to fall off the pier and cause the allele to go to fixation. Okay, so we've seen through computer simulations and mathematical exercises what should happen to populations, but just as before, it's always nice to bridge that gap between the theoretical and the practical or the experimental. And Peter Bury did this with fruit flies, a fruit fly experiment, sort of a classic fruit fly experiment, Drosophila, back in the 1950s. And what he did is he created a bunch of different populations of Drosophila melanogaster, which are the fruit flies, and he was able to figure out the allele frequencies of these flies by a morphological property of eye color. And so he could distinguish between homozygous BW, that's the allele, individuals, heterozygous individuals, BW75, and homozygous BW75 individuals. And so he could create populations that had a 50-50 frequency, 50% frequency for BW75 and BW. So he did that with 107 different populations. And he said, okay, how can we experiment to see if in these, not just in theory, in mathematical theory, that we should see these populations go to fixation for one allele or the other, but how can we do it in an experimental setting? And so what he did is he took 107 populations. The frequency of the BW75 allele in each of those populations was 0.5. Obviously, then the frequency of BW was 0.5, so we can have that as our P and our Q. And then he would, with each of those populations, select out 8 males and 8 females randomly and start the next generation with those 16 individuals, or with those 32 new alleles that he's selecting from each of those populations. So then he's going to start 107 more second generation populations, grows those up, scores the allele frequencies. Again, it's a small population size, so it could be that of those 32 alleles he selected from the previous generation, that half of them are BW and half of them are BW75, but due to random selecting error, it might not be that. And then he would score each generation for the allele frequencies of both of those alleles. Again, then in the next generation, generation 3 or generation 2, he would select 8 males and 8 females, grow them up, look at the frequencies, do it again and again and again, and he charted that for 19 different generations. So here are the results from each of those 19 generations for 107 different populations at each generation. So if we look at generation 1 on the right, they're just plotted frequencies from 0 all the way to 100%. And so you can imagine our parental population, if we would have charted that, we would have had a big bar right at 50%. We're looking at the BW75 allele, and you'd have 175 populations stacked up all at 50% because we set it up that way. If we look at the first generation, we see that distribution, and we can see it in higher resolution there on the right. We can see that the vast majority of the populations are indeed at 50% or around 50%, but we do get some deviation. Even in just one generation, we have some, if you look there on the left, that are all the way down there, just below 25%. So in one generation, simply due to random sampling error, you had one of the populations go that low. You have another population that's up there maybe in the 70s or so. And that's sort of the equivalent of the random act of flipping a coin 5 times. Well, you could calculate the probability, but we'll just say flipping the coin 5 times and getting all heads. So in one of the 107 population, that occurred. And that, shouldn't be too surprising to anybody that those random deviations can and do occur. Then as we look at the second generation and the third generation and the fourth generation, we see that the frequency of that allele are moving. They're sort of spreading out from that 0.05. And then by, it looks like the fifth generation maybe, there's a population that becomes fixed for the BW75 allele. Then by the 10th generation, you see down there, you see there's or even before that, 8th generation, you begin to see that there are populations that are becoming fixed at 0%, populations that are becoming fixed at 100%. Then by, let's say, 17th generation, we now see that the vast majority of those populations have become fixed for either the BW75 allele or the BW allele. And there's obviously, these are laboratory populations, so there's not really any apparent selective advantage in for any of those alleles. And so we see this dramatic change in the frequency of the alleles, or evolution. Remember the simple definition of evolution being the change in allele frequency from one generation to the next in all 107 populations. And the only thing that's acting on them is the sample size, or the population size is small in each generation, fairly extreme, right? Only 16 individuals in each generation. And over 19 generations, we see that the vast majority of them have become fixed or evolved to the point where you have 100% of the individuals either with BW75 allele or 100% of the individuals with the BW allele. This figure from the book shows the decline in heterozygosity. And so this is the average frequency of the heterozygous individuals in each generation, averaged over all 107 populations. So we're not looking at the individual populations anymore. We're taking the average of the 107 populations. So obviously in generation 0, all the populations were heterozygous. All 107 populations had all the individuals heterozygous. So you can imagine that 0, if you go to the 0, the first point would even be up there at 1. But then in one generation, we see that the heterozygosity drops dramatically, and in a different, if you think about it, when a heterozygous individual mates with a heterozygous individual, you will have a quarter of the offspring on average being heterozygous for one of the, or homozygous for one of the alleles, 25% or a quarter of the population homozygous for the other allele, and then 50% homozygous. And that's why in that first generation we come all the way down to 50%. That's just the genetics of it, right? But as we then go through each generation and average the heterozygosity in each population, you see a dramatic decrease in heterozygosity. This is because as genetic drift, as we mentioned before, is this steady march towards homozygosity. So as you march towards homozygosity, you dramatically decrease the heterozygosity. We can mathematically model that. We have the equation there at the bottom. You don't have to memorize that equation, but it's just saying the heterozygosity in the next generation is going to be due to the heterozygosity in the original generation, and then factored into that the population size. So as the population size goes down, right, that's going to approach that 1/2, and then that's going to make it so that your heterozygosity goes down. So smaller populations are going to give you a larger decrease in heterozygosity in that next generation. And here we have our experimental data there in the red, and then the dashed line there shows the theoretical prediction based on that equation at the bottom of the decline in heterozygosity based on a population size of 16. So what we see is our experimental population actually decreases heterozygosity much quicker than we had predicted. And so if we were to remodel that, not based on a population size of 16 individuals or 32 alleles, but I believe that one was 9 individuals rather than 16, that fits our data fairly well. And that can be explained by the fact that, maybe some of the other assumptions of the Hardy-Weinberg model are not being met. For instance, maybe not all of the flies, and I know this from the genetics course and working with Drosophila in the little vials, maybe some of them don't survive as well, or some of them die, or other things like that can happen. Maybe, sometimes even students will misidentify. I'm not saying this happened here, but there's other things that it could affect the population size. So you don't have, even though you're theoretically, or in the experimental design, you have 16 individuals. Probably not all 16 individuals reproduced equally or survived equally. Okay, but as you can see, the theoretical and so what we call that, we have our population size, which is our actual numbers of individuals in that population, and then we have another thing that's called the effective population size. And so in this experiment, we can see that the population size was 16 individuals, but the effective population size, the size that population is kind of acting as in a genetic sense, was 9. So not only do we have a lot of experimental studies that look at genetic drift, we just looked at one of them, but there's also other studies that researchers and teams that go out into the field to look at the effects of genetic drift on populations. One of the sort of classic studies was one done by Alan Templeton and his group from Washington University in St. Louis with collared lizards in the Ozarks of Missouri. Here you see a collared lizard that may look familiar to you. This is actually an eastern collared lizard, Crotophytus collaris. It is one of 2 species of collared lizards that we have here in Utah. The other one is a Great Basin collared lizard. But that kind of lizard, if you were to see that, you would say, oh, that lizard is probably associated with some desert, like it is here in Utah, either with the Great Basin collared lizard with the West Desert, but then down in southeastern and southwestern Utah, we also have the collared lizard species, but in our sort of xeric desert environments. And so if you go into Missouri now is the Ozarks in Missouri are full of oak hickory forests, but you find occasionally populations of collared lizards there. And so Alan Templeton and his group said, okay, why are these collared lizards here? What is the geological history, right, or the biogeographical history of the area? And can we use this as a natural sort of experiment to test the effects of genetic drift? So if we look back in history, what we find is that the Ozark Mountains in Missouri, which I've put a white triangle there in the state of Missouri, roughly the position of it, 4 to 8,000 years ago, it was covered by a desert that connected with the Southwest Desert. And so you can see where it says Southwest there, that's roughly the Southwest Desert. So you had a desert extending through Texas, a bit of Arkansas and Oklahoma into Missouri. And what you then had after that, after about around 4,000 years ago or whatever, you had the hot period ending and the desert retracting back to where we find it here in the Southwest. As it retracted across Missouri, it was then filled in with that oak hickory forest. And so what you see in Missouri is you have these rock outcrops, and in these rock outcrops, we call them glades, that's exactly where you find those eastern collared lizard populations. And so now the sort of geological history sort of explains, one, how we get those collared lizards there, and two, now from our evolutionary biology perspective, we can say, okay, at once it was sort of this big desert. There was probably a lot of connection between the different members of the eastern collared lizards, right? The populations and individuals in the populations. And so it was a big population. Then as the desert retracted, it left behind these small island populations on these rock outcrops in the Ozarks. And, not in all of them, but in a lot of them, the collared lizards that are associated with the Southwest Desert have remained there ever since. And so you have one large ancestral population now we being sort of fragmented into a number of smaller populations that inhabit those rock outcroppings. So what Templeton's group predicted is that if these populations are now relict small populations from this larger population that now for the last 400,000 years, not 400,000, 4,000 years had been affected by genetic drift, that they should find a number of alleles in these populations that had gone to fixation, just like we had seen with those experimental fly populations, and that the different populations should probably go to fixation for different alleles. There should be sort of a random distribution of which allele went to fixation in those different populations. And so what they did is they scored them for 3 different types of markers: a protein marker that we call an allozyme. An allozyme is just a protein run across a gel, electrophoresed across a gel. And if you see there, you look in the bottom right-hand corner, you see MDH, that's the protein, small protein versus fast protein. So there are 2 different kinds of proteins, one that ran fast across the gel and one that ran slowly. So you could score them for one of those 2. For the mitochondrial DNA, there are a bunch of different haplotypes, A through D. And then he also scored ribosomal DNA, and we had 3 different types of ribosomal DNA markers. And so what you see in that pie chart is that, say, for instance, there on the upper left, you have if a population was marked as blue, that means they were fixed for the fast, or they had individuals. So let's say, for instance, let's just take a row one, for example. So that blue one there on the bottom, it's all sort of that light blue colored FC1. And so what that said is when Templeton and his researchers went in there, scored them for these markers, they found that all of the individuals had fast allozyme, the fast allozyme. They had the C mitochondrial haplotype and a type 1 rDNA marker. And so that's what that pie chart shows you. And so then if you look at the distribution throughout, the upper one is sort of the large scale map, and then the Tom Salt Creek is the sort of a finer detail of one little section in there. But what you saw is throughout the populations, most of them were in fact fixed for each, a combination of each of these 3 markers. And that there seems to be pretty randomly distributed throughout that. And here we're looking at 3 different markers. So if we took those all individually, we'd probably even see all of those markers individually. Probably even see more populations that are fixed for those things. And so the natural populations confirmed sort of what we had predicted with our theory, and not only with regards to alleles going to fixation, but some other researchers, along with Templeton's group, went in and looked at levels of heterozygosity, and they wanted to check and see if, in fact, drift was the main thing that would explain this. You should see the larger populations having a little bit more heterozygosity than the smaller populations. And they did that by looking at these measures of polymorphisms and allele richness. It just sort of gives you an idea of the genetic variation. So the larger population should have a little bit more genetic variation. Their genetic variation should not have been affected as dramatically as the smaller populations. And in fact, they went in there. There's a figure in the book. They went in there and looked at that, and they confirmed that to be true. So this is also important for obviously for conservation genetics, for endangered populations. Is that these smaller populations are, when drift affects these smaller population, it decreases the genetic variation, and that decreased genetic variation makes those populations less likely to be able to adapt to change, which affects their survivability, and it also causes higher portions of the population to be homozygous, which homozygosity can also have deleterious physical repercussions. Health repercussions. Another example of when sampling error can cause a dramatic change in allele frequency is a phenomenon known as the founder effect. So the founder effect is sort of the random sampling error that comes when you take a subset of a large population, so a founding or colonial population from the larger population, that separates itself from that larger population and starts a new population somewhere else, isolated from the original large population. You can sort of think about it in historical terms as, say, for instance, in European colonization. So if you look at European colonization here in the Americas, say the Pilgrims, right? You have a small group of English folks coming over and starting a colony here in the Americas. So in that small population of Pilgrims, do you think that they carried with them all of the genetic diversity from the continental population? Of course not, right? And so you have this small fraction of the parental population that comes and starts a new colony population, and that's what's known as the founder effect, and that can dramatically increase the frequency of an allele. Not only that, but typically those populations also will be small and will grow through time, but as they grow through time, their numbers may be larger. Their numbers may increase in time, but their genetic diversity isn't necessarily going to increase because, barring any sort of migration into it, you have to rely on mutation to generate more genetic diversity, or mixing between different mating pairings. But you also have a small population, and so those populations are going to also be affected by genetic drift. And as we just learned, genetic drift is a drive towards homozygosity. And so what ends up happening is that sometimes you'll have a founding population. In that founding population, you'll have some individuals that maybe are carrying as in a heterozygous state, some sort of deleterious recessive allele. But then as those populations grow, and in subsequent generations, you see through drift and through non-sort of random mating, you have this march towards homozygosity, and these recessive deleterious alleles pop up in subsequent generations. These founding populations also can oftentimes be isolated by geography, but also by culture. And so here's a list of a bunch of different populations that were founded by a small number of individuals and remain fairly isolated. And in doing that, you have this high frequency of these conditions that sometimes are deleterious recessive alleles, sometimes not, but you get a much higher frequency of these conditions in these populations than you do in continental populations or in larger populations. And so you have a high frequency of such things as albinism with the San Blas indigenous folks in Panama. You see that there. You see with Amish populations in the United States, they suffer from a high incidence of Ellis von Creveld syndrome. You see Tay-Sachs disease in Ashkenazi Jews, a high incidence of that, and you see that repeated over and over again. So we have all of these natural sort of experiments where we say, okay, these are the real life sort of results of a founder effect, and followed by genetic drift, and then also by necessity, because they're small populations, you have a mating system which makes it so that you have a heightened probability of mating with a relative, and so you also have inbreeding in these populations. Founder effect also reduces total genetic variability, because as you have a small sample size from a larger population, you're not necessarily going to sample in that small population all the alleles that are present in that population. So you lose that genetic variability. A nice example of that are silver rays off the coast of New Zealand, and different islands off the coast of New Zealand, where you can actually see we have a historical knowledge of the silver rays colonizing one island and then later colonizing another island from that first island, and then in sort of in a stepwise fashion. And we can actually look at the genetic variability in each of those colonizing events, the founder effect in each of those colonizing events, and see in sort of a natural setting the reduction of the genetic variability. So here you have the dates of the different colonizing events: 1830, Tasmania to New Zealand South Island. Then you have range expansion within the South Island, and then a few years later going out to Chatham Island, to the North Island of New Zealand, then up to Norfolk Island. And so here we have a nice series of dates, historical dates, and different colonizing founding events. And you might imagine that with each of these subsequent steps down a colonizing event sort of cascade, what would you expect would happen to the allelic diversity at each of these times? Because you're taking a small sample of the previous population, and that small sample is then finding the next population. Their population sizes are going up, and then you're going to have a small sample of that one, go to the next one, and go to the next one. So what would we expect? We would likely expect genetic diversity to decrease with each of those colonizing events. And that's exactly what we see here. These are the results, and what we see is for each of the different colonizations, we have a decrease in the genetic diversity. So again, a nice natural setting, natural population to do evolutionary experimentation and test whether our theoretical predictions coincide with what we actually can infer from the real world. Another phenomenon that is very similar to the founder effect is the bottleneck effect. And the bottleneck effect, so the founder effect, you're taking a small sample of a larger population and then finding a new population with it. Bottleneck is very similar, but what's actually happened is your original large population, some sort of catastrophic event happens, and you get a dramatic decrease in population size, where you have a whole bunch of individuals, and all of a sudden only a few individuals. And then those few individuals, through time, the population grows to the original population size or somewhere close to that. or even just bigger. And so here, the XY axis, population size is on the Y axis, time on the X axis. So you see at A, you have a population crash. So you have a dramatic decrease in the population size. B then is a small sample of what the original population size was when you have that dramatic reduction in the population sizes. That's going to do all of the same things that the founder effect does. It's going to decrease your genetic. You're just going to decrease your genetic diversity, your heterozygosity, and increase your homozygosity. And then through time, maybe that population will grow, and may even eventually become the same size as the original population. So population at A and C have the same numbers, but based on our evolutionary theory, we would say, okay, but even though those populations may look on the surface to be very similar, their genetic diversity is going to be very distinct. And we do have a number of examples of populations going through these bottleneck effects and the results of that all over. One example of that are cheetah populations. Cheetahs went through a dramatic bottleneck effect around 10,000 years ago, where their populations nosedived. In fact, the population here in the Western Hemisphere, in the Americas, we used to have cheetahs here in the Americas, but they went extinct. Likewise, you had a dramatic decrease in cheetah populations in Africa, but they didn't go extinct. Instead, they just went through a bottleneck event. And what we see is that then cheetah populations rebounded somewhat in numbers, but that bottlenecking so reduced the genetic variation of the cheetah population, the species, is that you can actually take skin grafts from one cheetah to another cheetah, and they generally do not reject those skin grafts because they are so similar. There's so much similarity between cheetahs that have now been derived from that bottleneck event. So that's a very dramatic example of losing genetic diversity, and then even over 10,000 years, really not gaining much of that genetic diversity back. So the take-home message is that genetic drift, founder effect, and bottlenecks all have pretty much the same result: reducing genetic diversity, a march towards homozygosity. And this is all a result of a violation of that one assumption of the Hardy-Weinberg model, and that is the infinite population size. These are all results of small population size. Okay, so we are finally arriving to our last discussion of the Hardy-Weinberg model assumption violations, and that is the random mating assumption. Random mating obviously is having an equal probability of one individual, any individual in the population, to mate with any other individual also in the population. So non-random mating is sort of not doing that. And generally, when we talk about non-random mating, there are a couple of different types of non-random mating that we have sort of identified or labeled. One is positive assortative mating. That's when you mate with an individual that is more similar to you. And an important thing to think about when you're thinking about mating is that we are not going to either be positive assortative mating or negative assortative mating. Negative assortative mating is when you mate with individuals that are different from you. You tend to mate with individuals that are different from you. You don't have all your characteristics line up as positive assortative mating or negative assortative mating. It is sort of characteristic dependent. So an example of that is with positive assortative mating, you can think, okay, even imagine if you have a partner, girlfriend, boyfriend, wife, whatever, think about some sort of aspect. So are there characteristics that we tend to mate in a positive assortative sense? And we can see that, yeah, that's generally true. We think about height. So do tall people generally pair up with other tall people? Or is it randomly distributed tall people with short people, medium-sized people, etc. No, what we see is that tall people generally tend to mate with tall people, short people with short people. We can even see that within ethnic groups. So does an African American tend to mate with and pair up with another African American, or is there an equal probability that an African American will mate with or pair up with any other ethnic group? And the answer is obviously no. There's a higher probability that an African American is going to get with an African American, or a Mexican American is going to get with a Mexican American, or maybe even more generally, a Latino with a Latino. So for a lot of aspects, for a lot of characteristics, there is positive assortative mating. But you can even think of religious groups. Do members of the LDS Church tend to get together with and mate with other members of the LDS Church? Is there equal probability of somebody who is in the LDS Church to mate with somebody within that church or without that church? And so these are all examples of positive assortative mating. Negative assortative mating is when you tend to mate with people that are different than you. And so there are a number of examples, sort of the opposites attract, but even on a genetic level, the sort of the best example of this is the major histocompatibility complex. Genes, we tend to, if you mate with somebody that has very different major histocompatibility gene alleles than you tend to have offspring that have higher fitness, have better immune systems. And so this is an example of negative assortative mating. So, you could be mating with somebody in a positive assortative sense on some characteristics, on a negative assortative sense on other characteristics, and even randomly. I mean, you might think, well, does anybody ever really mate randomly? And if you look at particular characteristics, you can say, yeah, that this mating occurred from the perspective of that characteristics in a random sense. So, for instance, those of you who have partners, think about check your own earlobes, and we have two general phenotypes for earlobes. Either they're attached or they're non-attached. And so you can check yours. If yours is attached, was that a characteristic that you said, oh, I want to go find somebody who also has attached earlobes, or I want to go find somebody who has unattached earlobes? Probably that didn't factor into it. That probably wasn't on like an eHarmony profile, that attached earlobes versus non-attached earlobes, or the way that your hair turns, your calic on your scalp turns. Some people have calics that turn clockwise. Some people have them that turn counterclockwise. That also was probably not a characteristic that you thought about when you were deciding on a mate. And so with these characteristics, we actually are mating randomly. Another type of non-random mating that we talk about a lot in evolution, but then also in genetics, is inbreeding. So inbreeding is when you mate with a close relative. Or semi-close relative. And inbreeding is what we're going to talk about mostly. So we're just going to use inbreeding here to sort of look at the assumption, what happens when you violate that assumption of non-random mating by having lots of pair matings with close relatives. And this is important in things like conservation genetics, or in zoo populations of animals and things like that. And so there's a lot of research that has been done on sort of the theoretical background of inbreeding. So that's the one that the book talks about, and that's the one that we're going to talk about most here. Okay, so let's just look what happens in an example of sort of extreme inbreeding. And that is with selfing. And so some organisms are able to self, like basically mate with themselves. We see that a lot in plants, in snails, other types of organisms like that. And I want to point out that, I guess I'll sort of show it to you through the example. And so let's say we have a population of snails. They come in three different genotypes: A1A1, A1A2, A2A2, and all A1A1 individuals are going to mate with A1A1 individuals because they're selfing, right? All A1A2 individuals are mating with A1A2 A2. It's a cross between an A1, A2 because it's selfing A2 homozygous A2 individuals, the same thing. And so you can even do a little Punnett square. What happens when you have A1, A1 crossing with A1, A1? All 100% of the offspring are going to be A1 homozygous individuals, right? What happens? Same thing happens with A2 individuals. And so let's start down there at the bottom left corner, and let's just take a population where we have 500 heterozygous individuals, 250 A1 homozygous individuals, and 250 A2 homozygous individuals. Okay, so the A1, A2, or the A1, A1 homozygous and the A2 homozygous individuals, when they mate, they're going to produce 100% of their offspring are going to be just like them, A1, A1, or A1, A2. But when an A1, A2 crosses with an A1, A2, and it's, again, it's selfing, right? So draw the Punnett square, and what you see is half of the offspring are going to also be heterozygous, but a quarter of the offspring in that mating will be A1, A1 homozygous, and a quarter of them will be A2, A2 homozygous. And so in one generation, let's say we keep the population sizes at 1,000, one generation of selfing, you go from a population where you have 25% of your genotypes, or 25% of your A1 of your individuals are A1, A1 genotypes, 25% are A2, A2 genotypes, and 50% are heterozygous individuals, to in the next generation, you have gone from having 50% of your individuals as A1, A2 heterozygous individuals to 250. And so you have that. You've also gained in your A1 homozygous individuals and your A2 homozygous individuals by adding 125 individuals to the 250. That's where you come up with the 375, because a quarter, the 125 is the quarter of the 500 individual heterozygous individuals selfing. And so what you see is you still, if you look at the allele frequencies in that first population, you still have an allele frequency. A1 allele frequency is 50%, A2 allele frequency is 50%. If you look at the subsequent generation, the allele frequencies are the same. They're both 50%. But what you have changed is the composition of the genotypes. And that's what inbreeding does. It, kind of like some of the other things that we've talked about, is a march to homozygosity, but it doesn't necessarily, it doesn't do anything to the allele frequency. So it's not actually directly involved in selection, right, or in evolution. There's no evolution going on between those two, but what it does do is it changes the selective pressures, and so that in that way it can indirectly sort of influence evolution. Okay, so here this next slide shows you we take it out a few more generations. So here, when we take it out a few more generations, it's easy to see this march towards homozygosity, right? And so eventually, if you kept taking out, you would get to a point where there's less than one individual that is heterozygous, has a heterozygous genotype, and then roughly 50% would be A2 homozygous and 50% would be A1 homozygous. So that seems to be the effect of inbreeding, right? It is getting homozygous. It is again this march towards homozygosity, and so we're going to sort of quantify that in a little bit by estimating or calculating this coefficient that we call the inbreeding coefficient. So the inbreeding coefficient is how we quantify inbreeding, but to sort of get our heads around it, let's just think about what it is. And basically what it is a probability that an individual will inherit the same copy of a gene, the same allele from a common ancestor. And when that happens, the alleles come together in the homozygous state, and we call them being identical by descent because they were passed down from a common ancestor. And so let's see how we calculate that. So to do this, we have to work with pedigrees, which are just show sort of like genealogies, and they show matings between people and offspring that result from those matings over a number of different generations. And so here we have an example of a mating between. We have a pedigree that shows, if you look there on the top, let's just look at A, for example. The square represents a male, the circle represents a female. When you have a line connecting the circle with the square, that means that they got together and they had an offspring. And so up here you see that there is a female, right, that has had an offspring with two different males. So the male on the left, when the female had an offspring with the male on the left, the square on the left, the result was a female child. When the female had an offspring with the square on the right, the result was a male child. And then these two individuals, which would be half siblings because they share a mother, mated. They had a female daughter, resulted from that. And so this is a half sibling mating. So let's look at the common ancestor. So the first thing that you have to do is say, okay, here, down here at the very bottom, the sort of the grandchild, right? So we have three generations. The grandchild, she has a common ancestor, right? You can form what we call an inbreeding circle. And so here, the inbred individual there at the very bottom, right? They're inbred because they are the result of the matings between half siblings, and the common ancestor is the grandma. And so if we go back to the grandma, and we color code sort of let's just look at one particular gene, and we'll color code that gene, making one of the alleles the red triangle and the other allele the blue diamond. And we say, okay, what is the probability that that red diamond allele, right, gets passed down both to the daughter and the son of that grandmother, right? And so the probability that the allele gets passed down to the grandma to the daughter is 1/2. The probability that the allele gets passed down from the grandma to the son is 1/2, and that's what this is showing. Then for the son to then pass that same allele down to the granddaughter, it's also a probability of 1/2. For the daughter to also pass that allele down to the granddaughter is also 1/2. And so if you go through, these are 4 different events, looking at it from a statistical perspective or a probabilistic perspective. If we have 4 different events and we want to calculate the probability that all 4 of those events occur, we simply multiply them together. And so we have 1/2 times 1/2 times 1/2 times 1/2 equals 1/16. And so that's the probability that the alleles in that granddaughter will come together and form a homozygous genotype, because that same allele was passed down from the common ancestor, from the grandmother, or that those two alleles are identical by descent. That's the probability of it. And so that's 1/16. But we also have to look at the other allele, the blue diamond allele, and we can do the same calculation for that. That equals also 1/16. So the probability that inbred individual, the granddaughter, will have alleles that are identical by descent is it can be either one of the alleles or the other allele. And whenever you do hear or in probability, you add them together. That means you should add them together. So the total probability that the alleles will be identical by descent is equal to 1/16 plus 1/16 equals 1/8. And so that's the inbreeding coefficient, the probability that those alleles are identical by descent. Okay, that's sort of the math way of thinking. You can also interpret it in ways of saying, okay, there is a certain level for an individual, in a population. We can kind of say, okay, that individual has 60% of their genes are heterozygous and 40% are homozygous, right? What this does is there's that baseline level, right? What this does is it adds to that baseline level the probability that the individual will be homozygous for a particular allele by that fraction. So in this individual, this inbred individual, the result of half-sibling matings, she is going to have, on average, 1/16 of all of her genes. So, in humans, we have 20 to 25,000 genes. So a whole bunch of the genes that she has are going to be homozygous just because that same allele was passed down from that common ancestor. So it adds to the already underlying level of homozygosity. And so in this way, again, we see inbreeding, non-random mate, or inbreeding in particular, is this march towards homozygosity. And so you can draw out other pedigrees for other types of matings. We do that a lot in genetics class. I just want you to kind of understand what's going on here, and especially what the result is. The result is that you're increasing the level of homozygosity in populations. And this can be bad from sort of a from an evolution perspective if you have a lot of alleles in a population that are deleterious and recessive. Because what happens if you have a certain level of deleterious recessive alleles, if they're rare especially, there's a very low probability that they're ever going to get together and form a homozygous genotype, right? Because it's so rare that the vast majority of the time, if 1 in every 10,000 individuals carries that allele, then there's a very low probability that you're going to actually select a mate that also has that allele. So if you did, 1 in 10,000 chance that you'd select a mate that has that same deleterious recessive allele, because you're both heterozygous individuals. Neither of you are affected by it, but then if you do get together with that individual, then you have a 1 in 4 chance of your offspring having having that condition. So it's 1/10,000 times 1/4, so it's roughly 1 in 40,000% chance. But when you start looking at populations that are inbred, you have that added increase. So here, there's a pretty good chance that individual actually, in an inbred population, that those deleterious alleles are going to get together. And we know that in the human population, all of us have a couple dozen deleterious recessive alleles that are just sort of hiding out in the heterozygous form. We don't know about them, right? We carry them, but we're not affected by it because they're recessive. But when we start looking at inbred populations, what happens is those recessive alleles start coming together, and we can see the effects of that, and we call that inbreeding depression. This is a figure that shows the results of inbreeding and increased inbreeding. It's a study done on the bird called the great tit, and it looked at the number of eggs that failed to hatch in a nest. And so what they found is that as the inbreeding coefficient increased, so an increase in inbreeding coefficient means that there are matings between closer relatives, you saw an increase in the number of eggs that failed to hatch. This is part of inbreeding depression, and there's lots and lots and lots of examples of this, and it's a fairly common phenomenon, right? So we're done looking at all the assumptions. Before we end, we're going to go back and look at the greater prairie chicken and say, okay, what are the things that we've learned? What can this help to explain what is going on with the greater prairie chicken populations in Illinois? So just as a reminder, the Illinois greater prairie chicken was once a very abundant bird in Illinois, but then by the mid-1990s, it appeared to be destined for extinction. And like a lot of other endangered and vulnerable species, the prairie chicken's worst enemy had seemed to be habitat destruction. You remember we discussed the still plow. Before the introduction of the still plow, the prairie covered about 60% of Illinois, but now there's less than 1/100th of a percent that remains. But even with that dramatic habitat destruction, it seems that it wasn't the prairie chickens' only problem. If you remember, in the 60s, conservationists established some prairie chicken reserves and worked to restore and maintain that prairie habitat. And these efforts appear to be working. If you look at that chart there on the bottom left, going from the mid 60s to the 70s, the prairie chicken populations seem to be rebounding. But then we see that success was fairly short lived, and then they begin to nosedive. And so we, assume that something else was kind of going on, but we didn't really know what. And we alluded to the fact that possibly our learning about our discussion of migration, genetic drift, and random mating could maybe help give us some tools to answer why. And so researchers and scientists at the time hypothesized, they started to think about it, and they said, okay, let's make an hypothesis. And the hypothesis was sort of the following. The destruction of the habitat did two things to the prairie chicken population. First, it reduced the bird's population size, right? It dramatically reduced the population size, and we can see that in the graphs. But second, it also fragmented the population. By the 80s, the few prairie chickens that had survived in Illinois were trapped on these small islands of prairie in a sea of farmland, and that isolated each of these populations from each other. So when you have these small populations with little or no gene flow, this is the exact sort of setting where genetic drift is most powerful. And the genetic drift could result in the fixation, especially in dramatic genetic drift. Even if there's a deleterious allele, sometimes drift can even cause those alleles to become fixed in a population. And so if a bunch of alleles become fixed in a population, that directly reduces the fitness of the population, right? So with that reduced heterozygosity due to drift, we also have these small populations, and in these small populations, the birds that remained were, sort of forced to mate with individuals that they were probably related to, and so we get this double whammy. We get small reduction size drift happening in the small population size, right, with that dramatic reduction of size, but then also now you have just these isolated little populations, and so there's not random mating. They end up having to, by necessity, mate with close kin because they have no other choice. So they kind of fell into this extinction vortex, right? And that's exactly what we saw with the decline. But researchers wanted to go out and say, okay, do we see inbreeding depression happening? And so we have random fixation of these deleterious alleles possibly in the populations. We have these populations, small populations, that are also inbreeding. So we need to go out and actually measure and see if in fact we see inbreeding depression in the population. If you look there on the bottom right-hand corner, we see that's exactly what they found when they were looking at the percentages of eggs being hatched. If you look, the normal sort of percentage is in the upper 90s. And if you look at prairie chicken populations outside of this state of Illinois where they're larger, or if you look at them pre-1930s or pre, still plow, we have data that suggests, okay, this is sort of the normal. This is 95, 96% of sort of the normal. But you see in the 80s, it begins to drop, right? Even after the growth in the 70s, all of a sudden it begins, you look at late 70s to the beginning of the 80s, it drops down to 80%, and it just sort of continues on this steady drop till in 1990, the percentage of eggs being hatched were all the way down to 40%, Okay, so these data do seem to suggest that in fact these populations were suffering from inbreeding depression that is affecting the population. So again, so it's beginning to make sense to us. But we should also say, okay, we have this theoretical sort of hypothesis, right? It looks like it is inbreeding depression, but also we know, we mentioned a whole bunch of times that there's this march towards homozygosity or reduction in genetic diversity. So scientists would also want to confirm that, test that. And so they did that. They went, they looked, and they scored about the populations in Illinois for genetic diversity and compared that with other larger populations outside of the state of Illinois. And in fact, they found that there was a dramatic decrease in genetic diversity in these Illinois populations. So it does seem to indicate that all of these things that we kind of hypothesized were going on, and these aspects of population and evolutionary genetics, what we've been learning about, actually, did seem to predict and did seem to help explain this phenomenon that's going on in nature. But the flip side too is like, okay, so we can understand what's going on, but now because we understand what's going on, we can also possibly help use our knowledge to help this population out, to help cure the problem. And so in the 90s, that's exactly what researchers did. They said, okay, well, they're suffering from low genetic diversity, inbreeding depression. Let's get some prairie chickens from populations, larger populations in Minnesota and Kansas and Nebraska, and breed them in and introduce them into the populations. And we see from 1990 on, we have an increase not only in the population, rebounding in the population sizes, but also a rebounding in that inbreeding depression, with a percentage of eggs hatched. They go all the way back up to the 90, and even there, it looks like there's one that's even up to 100%. Okay, so these things matter. These theoretical things, yes, some of them are sort of mathy, and maybe not that interesting, but they have real-world application, and this is a good example. The real-world application of the stuff that we're learning about evolution, about evolutionary genetics and population genetics in the last two chapters, that can give us some real-world solutions. So to just sort of wrap up the last two chapters, we're looking at the Hardy-Weinberg model, how we could see patterns resulting in the different violations of some of the assumptions that went into that model. Overall, what that model did was just said, okay, the composition of genotypes and alleles, frequencies, and things like that from one generation to the next is just a random sampling, right? In order to make it that way, we had to assume a bunch of different things. And here I've just gone ahead and listed the 5 different things that we talked about: selection, mutation, migration, genetic drift, and non-random mating. And now we have a little bit of background, but also on each of these assumptions and the importance and things like that. But even more, I think another sort of take-home message from these last 2 chapters is that the pursuit of doing research in evolutionary biology, it's not just, in the beginning, Darwin's days, maybe it was just like, oh, this is how things could have evolved, et cetera, and it was just sort of creating a narrative. But what we see now is that the research into evolutionary biology is very predictable. It's quantitative. We can go out, we can make predictions based on sort of our hypotheses, and we can go out and test them not only in the experimental settings, but then also in the natural world.